Good morning. So this is week 3, and we've been talking about faith and evolution and how they go together. So today we're going to talk about irreducible complexity. I didn't plan on it, but I decided that we need to hit that, in as well as mutations and their power. So there are cracks showing in Darwin. It's clear not everyone in science is happy with the theory. It's losing some of its luster. Slowly, but there are some key people, and Lynn Margulis is an example. She's always been a critic. She has since passed away, but she's a member of the National Academy of Sciences, and she said, I was taught over and over that accumulation of random mutations led to evolutionary change, led to new species. You know, that's the basis of Darwin, right? I believed it until I looked for evidence. Now, she is not anything close to what you might call intelligent design proponent. She's clearly an evolutionary person, but nevertheless not happy with Darwin at all. Another bigwig, David Galertner, he's a famous pioneer in computer science. He created some of the key tools that allowed the computer age to begin back in the 80s and 90s. Currently a Yale professor, very respected. People call him a polymath a lot, and I had to look that one up. It means he's good at a lot of things, and he even creates, artwork that sells for good money. Back last year, he made public his recent rejection of Darwinism as a mechanism of increasing complexity of life, and this sent shockwaves throughout academia when he came out. He said, like so many others, I grew up with Darwin's theory and had always believed it true. Then he said, no longer. The origin of species is exactly what Darwin cannot explain, which is ironic because that's the name of Darwin's book. And he rejected it really based on a couple things: probability, the evidence, and really basic logic. He made clear he had no other philosophy to fill the void. He still had issues with intelligent design, but it was the writings of intelligent design proponents that convinced him of the utter failure of Darwin to explain the evidence. In particular, Stephen Meyer's book, Darwin's Doubt. He said, it convinced me that Darwin had failed. He cannot answer the big question, end quote. And few open-minded people will finish it with their faith in Darwin intact. And I like his use of the word faith. So why do highly intelligent, non-religious people have issues with Darwin? Well, we saw in week 1, there's an extreme time probability issue with spontaneous life arising. Then we saw last week the fossil record contradicts many claims of Darwin. Really, there's a lot of evidence that's missing at the scene of the crime. And fossils are limited. It's for forensic evidence. We're looking back, trying to interpret, what happened based on the past evidence. Today, it's different. Today, we're looking at mutations. They're the source of change. We can see those happening today, and we must understand their full power, because that's where the building blocks come from for change, is mutations. And we're going to see that they're very powerful indeed. But first, we'll talk about irreducible complexity. It was mentioned in week 1, mentioned in week 2, so I thought I need to look a little bit more into it in week 3. So here we are, and primarily we're going to talk about the flagellum, bacterial flagellar. This little thing here. This is a protein that whips around in the liquid and drives. It's like an outboard motor. It's like a highly complex outboard motor. When he came out with this book back in '96, this book here, Darwin's Black Box, it raised a new level of objection to spontaneous generation. It's a new level, and I'm going to explain that. And it caused a big stir in science. He gave multiple examples of what he claimed was irreducible complexity. And what is that? Well, if you look at a cell, it has certain kind of machines. They look like machines. They're made up of many different discrete parts. And if you remove one part of that machine, it doesn't work as required. So that means you can't build it one piece at a time. It has to be there, be ready to go, and fit perfectly, or else it fails. So the implication was clear to Darwin. Darwin must build layer by layer, piece by piece. Thus, it can't explain these machines. It was his contention. There was a trial in 2004 when one school district tried to introduce the thought that Darwin might not quite be right. But then there was a trial, literally, that highlighted irreducible complexity, Behe in particular. And eventually, the school board got rejected by the judge, and and then you could hear a collective sigh of relief in science, quote unquote. And you know, people thought, well, there we just showed that irreducible complexity is a dead end to worry about. Let's see if that's really true. I hope you saw the video. This is an excellently made video. It's got everything in it that you can imagine. You know, judges battles and this and that, back and forth. It shows the flagellum working, and it's amazing how it works. I'm not going to show a video. I'd like to, but I'll describe it for you. This is it. This is it right here, and this is the protein I was talking about. This whips around in the media and propels the bacteria. In human terms, it would be over 200 miles per hour. So this thing whips around. This is buried inside the cell. So this thing turns. First of all, it's made up of 30, this part here, the construction materials, 30 different proteins. It moves up to 100,000 RPMs. Now, if you know anything about motors, that's ridiculous, purely ridiculous. It's got a rotor, a stator, a drive shaft, a U-joint, a bushing, bearings, and a propeller. It can stop on a dime. It can reverse direction with a quarter turn from here to here. A quarter turn, it stops, reverses, and with near 100% efficiency. And it's hardwired into the bacteria, so the bacteria can control it and go where it wants to go. When it senses food over there in the corner, it can move in that direction. It's really very, very high tech in low life. And those two videos I just showed you, one of them shows it being put together. It's incredible. Now, Kenneth Miller, he objected to this, and he thought of giving Darwin a black eye. He came out and said to the judge, no, this isn't possible. He was their main witness. The pushback really was, during the trial, another machine found in nature, bacteria, was identified, was brought forward, and that's the type 3 secretion system, this guy right here, which is really a needle that the bacteria uses to pierce other bacteria and shoot something in or take something out. I'm not sure. But he argued evolution could have built the flagellum by incorporating existing parts elsewhere, putting them all together with this as the base, and building the flagellum. And I say, really? Piecemeal? Yeah, evolution would need to co-opt existing parts and build the flagellum motor. And where are these parts? The secretor has 10, flagellum has 30, basically 20 that it doesn't have. And you need not just parts, but parts ready to go to work, the right shape, the right size, and then you need another machine to bring them all together at once. And it needs to start working at time 0, you know, use it or lose it. Not to mention there's probably another 40 proteins, I'm just guessing, you need to regulate its construction. You know, when do you bring this piece and that piece, and how does that fit together? Yeah, the control mechanisms, how to turn it this and that way. You need other proteins to control it. But I always hear this, you know, Tim, well, it could happen, you know, it could happen. Well, it's great. Go in the lab and demonstrate, you know, just show a tiny little minor step in the right direction. That's what we want to see today, complexity. Show us nature building complexity. Imagine a mousetrap. There are many different pieces to a mousetrap. Which piece can you take away and still have a functional mousetrap? I would wager a bet you can't take away any piece. This is a very tightly knit machine. This is irreducible complexity. Imagine, you know, requesting these pieces and only given a very general description. You know, I need a metal bar with a hook on the end. I need a spring. You describe these pieces, and someone goes in the barn and starts looking for them. He might ask you, a metal bar, well, how long do you want it? And I'm like, whatever, you know, whatever. Just give me a metal bar. Now imagine he brings these parts and he puts them in a pile in front of you. You know, you think there's going to be a mouse trapped inside that? Well, of course, that's silly. Now imagine applying intelligence and trying to assemble the trap. You know, it's not going to work because the parts weren't designed to fit together as a whole at the beginning, at time 0. And even if you had the instructions, it's not going to work. So that's what we're talking about here.
Tim, if I can interject here, there are methods that people have proposed for how this could have happened, and it's not quite here. For one thing, you have lots of diversity within the flagellar structure, in terms of structural diversity.
Mm-hmm.
So there's thousands of different ways that these motors can come together.
Come together. No, you just changed the subject, really. You said they exist,
No,
then they
yeah,
have to come together. That's a different animal, really.
I'm using come together a little more colloquially here.
Uh-huh.
And also, not all of the parts are needed for every version of this. A lot of them exist without certain proteins, in particular the ring structures. So and there,
Has anybody proposed trying to construct one of these piecemeal? You know, all the information that's there is incredible. So send me that information.
okay, and this, I mean, there's quite a lot here, in terms of stuff, and the fact that, and with a lot of the irreducible complexity, the problem is that we find out things become less and less irreducibly complex over time in terms of our understanding of them. So yeah.
I mean, you got to imagine this thing being built piecemeal. You know, you're asking for extreme luck to happen.
But
Beyond
a lot of these proteins are already in the cell doing other things too.
where did they come from? I mean, no. Well, okay. Well,
a lot of them are, and a lot of them aren't truly unique. So the 30 aren't truly unique. A lot of them are homologs of each other. So a lot of them are very similar proteins to each other. So
okay,
you would expect them to be in the cell.
We got to get into the mutation today and what it takes us to select, for example, a homolog, you know, one that's similar, but of course has several differences in it. And so I would propose, I mean, I talk to people, and for example, one guy in particular, he said, yes, the human eye is extremely complex, you know, beyond imagination. But there are creatures, he said, with light sensitive parts. And then he said, I can see how that could lead to an eye. Well, that's typically how evolutionists look at things. You get a part here, a part there, and then there's this incredible empty space that they just blush over. And I would say, wouldn't a vastly more likely scenario be that the flagellum degraded into a secretor? And what's more highly probable? You know, randomly gaining immense information or randomly losing immense information. And that's really the second law of thermo. It's entropy. If you leave something by itself on its own, it's going to
in a closed system.
Well, there are no true closed systems, right? The whole universe.
Well, the universe is a closed system.
So if it sounds so easy, great. Go in the lab and demonstrate it. Just propose some simple experiment and start to build complexity. That's what's missing in all the evolutionary arguments. No one's taking the challenge.
Well, the whole thing with the whole thing with intelligent design, though, is that you can't experiment. You can't prove these experiments either. You can't.
I'm not proposing intelligent design today. I'm proposing that Darwin's theory is extremely inadequate, and people gloss over those inadequacies for why I don't know. It boggles my mind, and that's what I want to point out today. Is those things. So I'm not going to leave you with an alternative explanation today. Just leave you hopefully wondering why is Darwin looking harder and harder to do? As many people in science will tell you this.
I found it interesting on that video that you gave us to watch, is that one professor that touts Darwinism, he's actually put organisms in his laboratory and he's left them there for, what, 15 years now?
Yeah.
Hoping that they will turn into something else.
Exactly.
But so far, nothing's happened.
Well, that's going to be front and center of my talk today. Yep, that's definitely a big one. And then afterwards, and there's papers on this in 2015, 2016, that it was shown that this thing here is younger than this thing. This is older using molecular biology dating methods. This one is older. And then right after the trial happened, I mean, this is how it unfolded. Lawyers assured the judge that Darwinian processes could produce a flagellum. This is back, 2004. But a scientist who advised the ACLU, he had a different story a year later. He actually wrote a paper and proposed how to test it. And why? Because he said the flagellar research community has scarcely begun to consider how these systems evolved. Yet they were willing to tell the judge, don't worry, this could happen. And it likely did happen this way, with zero evidence that it could. So we know mutations have three possible ways. You can help a protein. You can mutate, this protein here or here, and it might help to break the lactose a little faster, for example, if this is lactase. And you can hurt it. You can imagine you could pop this thing in several places, and it won't work as well. It might even kill the protein. Or it might do nothing that you can actually detect. You might change an amino acid right here, and for all you can tell, nothing really happened. So the helpful mutations happen, but we don't really care. I don't care about helpful mutations. The gene might work faster or be more selective between two similar choices of sugar. These are rare things that we identify. When an impact to a protein is observed, it's like it changed and I can see that something happened, it's overwhelmingly bad. In lacking a good number for good mutations, people a lot of times will assume the bad to good is a 1,000,000 to 1 ratio. And I don't want to see good mutations. I want to see complexity happening, not old enzymes working faster. Let me fix this a little bit here. There we go. Must have happened. So, a classic counterexample is antibiotic resistance. I hear this one all the time. And I would say this is not only a bad one example, it's a sad example. Let's recall that, you have DNA, it's your code. And sometimes that code messes up. When a bacteria replicates, for example, instead of putting a G there, it puts a C, then you got a U, G, C in the RNA, and now it doesn't see the UGG that it was, which means tryptophan. Now it sees a different code and it says, oh, it's a cysteine. So it puts a cysteine there. So now you have a true, impactful mutation on your protein. And antibiotic resistance, for example, if this is your protein, something like penicillin here can attach to a site and block that protein. And all of a sudden, you can't use that protein because the active site is blocked. And now if you need that to live, you're going to die. If you get a mutation right here, a different amino acid, and now it can't bind to it, now you're resistant to that antibiotic. And how do you find these? Well, they occur naturally. If you have, say, 100 billion cells in a flask and one of them naturally mutated, how do you find it? Well, you add the antibiotic and you kill the rest, and the one is there to keep living. So that's artificial selection. The mutant was already there, and I applied something to get it out. And so I would say, an intelligent agent applied an antibiotic and selected an existing mutant. So this, I would say to the evolutionists, you don't pick this as an example of intelligent design to support evolution. It's kind of the opposite. It's intelligence at work. It's just like, well, what really will happen is the lone survivor has actually lost information, the ability to bind the antibiotic. It's still a bacteria. It's evolved, but just not up. It's taken no steps to be a more complex bacteria. The medical doctor by accident and the dog breeder on purpose, they do the same thing. They use artificial selection to find what's there, or to choose who gets to live and die. Another one is, go ahead.
I was going to say, but there are instances with bacteria of if you put, I think it's E. coli, that if you put it in citrate, it will develop the ability to consume citrate, and it's not natively able to do that.
Oh, wow. So it developed.
It takes it. What they did was they put it in a solution of glucose and citrate.
Uh-huh.
Because you'd have to at least get it through a couple generations anyway for any mutation to
Right.
happen. 'Cause, but
Okay.
they
Wait,
found that in
that's
about 40 generations.
40 generations?
In 40 generations, the bacteria started having specimens able to consume citrate. And it's just because they were mutations occurred allowing it to
Right.
express proteins that could eat citrate.
Okay. So it
But
got new proteins. All right. You know what? I think you're talking about Linsky was the first one to show that, wasn't he?
I believe that's the person.
I'm going to talk about that in detail here. Okay. Thanks for bringing it up. Hang on. We'll get to that in a minute. So another way is basically bacterial sex. You know, this bacteria transfers genes to this guy, and it gets the gene that's used to immobilize the antibiotic or kill it. You know, it's true. It's an improved bacterium, yeah, that can make proteins that can inactivate the antibiotic, but it's acquired nothing novel. You know, it's just like the other bacterium now. You know, we want to see Darwin demonstrate the ability to form complex structures. We want novel complexity. After all, Darwin said, you know, this process could build perfection of structure and co-adaptation. We're gonna look at the evidence for that in a minute. So I say, imagine, you know, a wide-eyed student ready to absorb all the proofs that demonstrate advancing complexity, and they would be disappointed to learn Darwin's most far-reaching theory that organisms can build tremendous novelty and complexity is still a postulate. I would say that. There's no lab test we have yet that shows, oh, look, this just got more complex. And that citrate enzyme example we're going to look at, that's one of the best ones that's touted now. So I say, for the love of God, just go in the lab and generate complexity. You know, start building something complex. And really, if you look at who's tried to do this and say, I'm going to put evolution on the line, very, very few people have tried to do this. Now, imagine if you did do it. Imagine if you could start slowly build something from randomly build something and say, oh, look, before it didn't have this protein at all, couldn't even see it. And now look, you would get a Nobel Prize in an instant. But I know it's hard. You know, evolution experiments are difficult, one might say. And you might say that, well, you need millions of plants or animals, you know, just to start the test, not alone finish it. And then you need tremendous time to observe it. You know, most research grants, what, 3 years, and then you're done. Close up shop, get some more new money. You know, it takes a lot of time. I get it. So Behe addressed a lot of this in his book he put out last year, Darwin Devolves. And he looked more at the molecular DNA evidence. But indeed, he said there is indeed a natural laboratory going on right now in Lake Victoria with the cichlids. It's estimated that the lake got filled up again 15,000 years ago. And if you go look, there are 500 unique species, these guys right here, in that lake. And I say they're unique, which means, yeah, they had to have evolved in that lake in this time frame. But we look, and there's no new developments here. And George Barlow, an expert on it, disappointed, says what seem to be major changes in appearance, these guys have evolved with little alteration of the basic plan. It means nothing new that I can get a Nobel Prize and win for. And this highlights a growing sentiment. Darwinian evolution is self-limiting. The same factors that make it work well on a small scale don't allow it to go very far. And this is not a bad example of microevolution. It's a very good example of microevolution. The fish is still a fish. You know, we wish we could have been there to watch it happen. You know, certainly suggests that any such lab work takes long amounts of time. So you might start to begin to sympathize with this problem. And so you might say, well, what would be the perfect lab experiment? What would be the ideal lab experiment? Well, you would run it without any interference in the evolutionary process. And you can't, as a scientist, go in there and tinker here and tinker there. No artificial conditions or barriers, no artificial selection. Just let nature do its thing, you know? And of course, be able to grab a sample every now and then so you can watch the genetics change over time, watch the mutations do their thing. This is a Darwin field day, my dream here. You got mutations, natural selection, and then whatever comes next, you just let it happen. And make the best organisms live, a long and prosperous life. As Spock would say, may the weak ones die the Darwinian death they deserve. That's Darwin, survival of the fittest. And I also want to see like 50,000 generations. That's a long time. And could I be there to watch the whole thing happen, unfold? Well, is this a naive, expensive, time-consuming, maybe a drug-induced dream that I'm having right now? Let me introduce you. Actually, I know I forgot to say no, no, because Mr. Richard Linsky here, microbiologist at Michigan State, he started an experiment in 1988. It's going on today. Today, they're working on that experiment. Some poor grad student has to go in on Sunday morning and do it. So that's really over over 1 million years in human years. This is a pretty good experiment. It started well before any thought of DNA analysis, the hope of doing that. And here's what he did. This is kind of what I do for a living right here. He took 12 flasks, put the food in there, you put E. coli in there, and you let them grow. And they grow 6 or 7 generations in that day. And then the next morning you go in, you take each flask and you add to this flask a total of 1% from this flask. So you would re-inoculate. You take out the ones that did the best, or a representative, which means the ones that did the best would be the highestly represented. So you take them out. You don't select. You don't say, I want you, and not you. No, you say everybody gets to go. Who wants to who wants to be a part of this 1%? And then you repeat that every day for 32 years, including today. So this is an awesome experiment. I call it the best experiment ever. No interference, no real artificial conditions. The bacteria are living and breathing what they want to do. The organisms, they're free to evolve. However they want to do, go do it. And you can go in there and you can sample it, yes. And is a true Darwinian outcome possible? Yes, the best growers win. The slow growers lose. That's survival of the fittest in its truest form. And Linsky, he would go in there, he would pull samples at every 500 generations or so and put them in a freezer. So this is a living fossil record. And I do this for a living. You can go back 30 years, that bugs that I froze, and there they are. I can make them live again, put them in some media, and boom, there they go. It's really cool. So it's evolution right before our eyes. So what happened? After a few hundred generations, the cells started to grow faster. Pretty cool. 2,000 generations, they were doing about 37% faster than the original ancestors. Some flasks grew faster than others. That's what we would expect. Random mutations at work. Each flask gets its own set of mutations, and it will help it or hurt it, however those mutations dictate. But they were all growing faster. Interesting. And by the way, doing 12 flasks, that's incredible repetition. You don't get that a lot of times in experiments. So you might do 1 flask and have some results, and someone will say, well, if you'd done it 12 times, who knows what you would have seen. Maybe you got lucky. I always say in science, it's once for luck, twice for science. And then you do it 3 times. After 50,000 generations, the cells only grew 70% faster than the original guys. So it peaked off, shot up really fast, and then kind of tailed off. So what happened? What was the cause for the faster growth? You could imagine the excitement that they felt. Were new genes constructed? Were old genes ramped up? But up until about 2000, you couldn't really do widespread analysis of these guys to see. You could do a lot, a little bit, if you knew where to look. I had one idea. All the 12 flasks, they noticed that they all lost the ability to eat ribose, which is a 5-carbon sugar. And so that means they had a degradative mutation. They lost the ability. And funny, by losing that ability, they noticed that it can grow faster. Interesting. Now it didn't actually grow wheels or mutate wheels on here, so don't get too excited. It just grew faster. And this is really an oxymoron. This is a phrase you haven't heard much a lot. People give examples of, but no one really zoomed in on this as what's going on inside of a mutating organism. And it's an oxymoron. It's degradative, but it's beneficial. That doesn't make any sense. Well, yeah, it does. It lost the ability to eat ribose. Now it can grow faster. That's the benefit. I mean, that's not a natural way to think, but this is what happened. So how in the world could destroying something lead to a positive result? You know, how? Let me give you a good example. Imagine you're a car, okay? And you get a certain gas mileage. Here it's 23.6 miles per gallon. And then someone puts a gun to your head, pardon the analogy, and says, you need to get better gas mileage or you're going to die. Oh, okay. So what do? Well, like the bacteria, you say, okay, what can I do to go faster? Well, what you do, the first thing you do, you get rid of the back seat. I don't need the back seat. I need to get lighter and get better gas mileage. Spare tire, it would be nice to have a spare tire down the road, but right now, I gotta go get better gas mileage. So you pitch it. You don't care about the future. This is evolution. You don't think about the future. You do what's good for you right now. Okay, the gun's still at your head, not good enough. Okay, windshield, you're gone. All the glass gone, the muffler gone. Okay, the car sounds louder, but who cares? I just got better gas mileage. Now I'm at 25.1. But you say, I'm gonna need this windshield, Tim, you know, a half mile down the road because there's a rock headed for my face. Well, sorry, we can't see that because we only care about the right now. And even though you need a windshield has definitely saved my life one time. You know, it doesn't matter. You only have one objective, that is grow faster, get better gas mileage. And that's what you do. That is the natural thing to do. And this is exactly what one would expect from a blind, aimless process. Evolution deals with the problem at hand. If there's a new problem coming in 5 seconds, then I'll learn about it in 5 seconds. You know, it might take me 10 seconds actually, but it certainly won't be now. It's like you're on a boat that's sinking. Nothing's sacred on that boat. Nothing's sacred. Even the gold treasure that you found 2 islands back, you toss it over. And so you might ask, why do bad things happen to good bacteria? Well, because they like it. And I won't even go into that right now. So let's fast forward to the 21st century. We can take a bacteria's DNA and read it so quickly. So what did Linsky do? He took all his generations, 500, 1,000, up to 50,000 generations and said, let's track the changes and see what happened. So they did 264 complete genomes, and there's 6 million base pairs per genome. So it's a lot of work. So the precision to identify key changes leading to higher evolved states now is unprecedented. So what was the full breadth of changes that these Darwinian forces impose upon these bacteria that can grow a lot faster now? Were new genes identified that brought forth entirely new functionalities, some tweaked up to be super, super efficient genes now. A little less glorious than that. 2 key classes of genes. Class 1, the same 15 genes in all flasks had point mutations, but different mutations. Basically, there's 15 genes in all of them, and those genes got damaged in different ways, which means the bacteria doesn't care how you knock it out, just knock it out. Just shoot your machine gun, random mutations, degrade it way down. And then there's 16 other genes shared amongst all 12 flasks, completely destroyed. Big chunks missing or big chunks added or whatever, not ever going to work again. Destroyed. Let me tell you what I'm talking about here. Imagine a gene. Here's your DNA. It codes for a protein, this guy. And this is the gene laid out right here, imagine. And there might be some ways to improve the function of that gene, make this lactose get broken up faster. But those will be very, very limited spots. And these things already work pretty efficiently as is. It's hard to improve something like that. Not impossible at all. It just takes a very smart person to do it. But on the flip side of that coin, there are many, many ways to hurt that protein. And these might be ways where you can do a mutation in here to result in a protein that is damaged. And then if you happen to add a base pair to this thing or take a base pair away, that's called a frame shift mutation. It shifts the reading on everything down this DNA or the RNA. And so you get complete gibberish. So there's many ways to do that, and you damage the protein. So it's easy to break. It's much harder to do something good with it. Of course, that's the second law of thermo. And here's an example I mean. Imagine these 2 point mutations pop up, just these 2, and that will damage your protein. Okay, And then in another flask, these 3 in green pop up and damage your protein. So the bug doesn't care how you hurt it. It just wants you to hurt it so it can grow faster. So that's it. 50,000 generations. Such market improvement in growth rate. Now, the granddaddy is left in the dust of these new generation, the 50,000 generation. They grow so much faster, so much relentless mutation, so much selection of superior strains. It's, after all, it's survival. And what do we get? All of the observed beneficial mutations, they work by either degrading or destroying the ancestor gene in all 12 flasks. Certainly, no new evolved capability, no added complexity generated in 50,000 generations. Not even an obvious improved existing gene occurred. I'm not saying that couldn't happen, okay? But what clearly dominated the landscape was decay and destruction. That's how they grew faster. Which way did he go? Darwin says you got to go up, my friend. You must go up. We saw in this the best evolutionary example ever. It raced downhill. Over 31 genes destroyed or greatly degraded. And these aren't real wheels. I'm reminding you that. No, it's just it grows a lot faster. It just chucked off a lot of stuff it didn't need. So it took genetic inheritance. It had genetic inheritance, and it traded it for short-term gain. Okay, and I say that loss is gone forever. There's no going back. That bridge cannot be rebuilt, and I'm going to talk about that later in a second. But here's that citrate thing that I think that started the whole conversation in the evolutionary world of citrate. I was a little bit unfair. Yes, I was a little bit unfair to Linsky. I said nothing new had evolved. Linsky, he claims very enthusiastically that I'm dead wrong. He put citrate in the fermentation medium, and that component was not intended to be a food source. But one day he looked and he noticed extra growth had occurred in one of the flasks. Interesting. This is cool. I mean, this is interesting. If you're an evolutionary guy like he is, something like that gets your attention. And indeed, E. coli had begun to consume the citrate. What is going on? Let's talk about how that could happen. The gene in question is citrate transporter gene. The bacteria make it, and it goes and collects citrate, or it allows it to go through the membrane into the cell when it's present in the medium. So when oxygen is present in the non-damaged condition, there's a control region here that blocks this protein from being read and expressed. So if oxygen is there, it doesn't get made. Here's an example. Here's oxygen, and here's the citrate not being consumed. But you take the oxygen away, which is easy to do. This little thing right here pops off. And now you can read this and make this transporter, and you can go get citrate. And so with no oxygen, now you're eating citrate. You see how it's going into the bacteria? That's my idea of it eating it. What Linsky saw was the organism could consume citrate when oxygen was present. That was different. It could eat it when oxygen was present, because that's how he operated his fermentations, always giving them oxygen. So let's review. In the damaged state, what happened was there was a new controller element that was placed right in front of the transporter. Now it could make this protein, whether oxygen was there or not there, whenever it wanted to. We call that constitutive expression. So oxygen's there, don't care. I'm going to be eating citrate. So now he can eat citrate. That's pretty cool. So the discovery was touted as having acquired a new functionality, which is great for the theory. That's great. But I say, wrong. It could always eat citrate. That's nothing new. What would have been really cool is now it could eat a sugar that it couldn't eat before, under any set of circumstances. All that happened was the control mechanism got destroyed. Now it's free to eat citrate whenever it sees citrate. That's the change. This is boring. This is not increased complexity. It's like my wife, she goes to turn off the light, and she hits the switch, the light's on, but it's again on, are you going to start thinking that the house evolved complexity because of that? No, she's going to say, hey, Tim, or sweetie, I got a job for you. And what do you think that job's going to be? Hey, Tim, something's broken. I need you to fix it. This is not upward. This is not building upward complexity. This is embarrassing to say that this is my best example of evolution happening. This is ridiculous. It already could eat. It already could.
Tim,
Right. Go ahead, Joe.
if I'm actually glad you turned back to this slide. Because you said earlier that Darwin says you have to go up. Darwin doesn't. I don't know what Darwin himself personally said on this matter.
He said you could build, perfect complexity, perfect structure, starting from essentially, ground zero.
Did he say? Well, for one thing, let's get back to the perfect part of that. But evolution doesn't say you have to go up. It says you have to meet your environmental needs.
Right.
And honestly, one of the things that I see is wrong, is a problem with the Linsky experiment and why you wouldn't expect to see much variation, is he didn't change the environment very much. So like that's a lab situation versus a real world. And I've had plenty of experiments because I've done both field stuff and lab stuff that if you do it in the lab, it all works great, but then when you go out into the real world, it's a lot different.
Well, I'm saying that condition, is something that the bacteria could experience. They have plenty of food, everything it wanted, oxygen, and so just grow. And we put a requirement on it. You must grow faster than your buddy if you want to stick around. I mean, that's survival of the fittest. I mean,
yeah,
Tim, can I interject? And the way I understand it is he created the perfect environment. He took away any stuff that could have interfered with it in a negative way. He gave them a perfect place to live, and he still didn't get the result he wanted.
Well, and okay, so two points. For one, making a perfect environment is part of the point. Is if you have a perfect quote unquote environment and you're only selecting for them to grow faster. Well, we could take a bunch of people and start selecting for who's the tallest, and that's only a couple genes. That's only a few genes by comparison. So yeah, people will grow taller with that, but that's about all you're going to get. And second, you don't get to choose the results you like or dislike. That's not really how science works. And
I agree with that, by the way.
What?
I agree.
Yeah, I know you agree with that. And I just wanted to kind of clear that up because even colloquially, I think it gets kind of thrown out that we like this result or not. And at the end of the day,
yeah.
I mean, I think there's even like a journal of negative results or something like that. But but it's not like I don't think it's ever like someone's worldview is shattered because they don't get the results they don't like.
Absolutely. I mean, Linsky's hardcore evolutionist to this day. I agree, and I'm just saying he is applying philosophy. I'm saying that the objective observer would say, you did the best example of evolution we have to date. Okay, you created a niche environment. It's artificial conditions, by definition, because it's in a lab, right? We're not out in there. But you did the best you could do, and you simulated the best you could, and you set up the organism as best you could to do its thing, and it did its thing. Okay, let's just look at the results and extrapolate like humans do, right?
Right. What I'm saying, I guess, is that in a real world environment, there's a lot more factors that can contribit to mutation.
And you would not be able to deconvolute those factors easily. That's one of the problems with real world experiments. That's why I deal with crops. You got droughts, you got hot spots, you got bad soil here and there. You got a million things to think about. So as a scientist, you try to scale it back, the things you can control. And with those caveats, you push go and you do your experiment, and then you explain the extraneous possible impacts later as you interpret the results. And that's what we're doing here. And I'd say this is not encouraging. I mean, at the very least, you know, this is a little discouraging to a hardcore evolutionist. I would have to think it would be. And it's to tout that citrate is an example of evolution. Man, come on.
Okay. I'm trying not to get irritated. But
You're doing a good job.
I mean, in terms of back, like learning new information and stuff like that, I mean, you probably know as well as anyone that viruses can influence genomes pretty effectively.
Absolutely.
You can introduce completely new DNA into a
Yep.
cell, and provided and when you have that, then you're able to make new proteins.
Yep.
Initially probably for that virus, but as we went with the type 3 secretory system and the flagella, that different proteins can be used for different things. It doesn't have, it's not a one-to-one that you have to use one protein for one certain thing.
It helps, but yeah, right.
And viruses are just one example of something.
They can bring in a lot of new genetic information, but then you ask, that's what you have to ask, where did he get his information and his information, and how's it all build up? And now I'm going to get into how powerful mutations are. What is their true powerful, power building, power bringing capability? And we're going to look at Michael Lynch, very famous, mathematical geneticist, is going to speak on this matter for it. So let me press on. But everywhere we look, we see things going downhill. This is new stuff. Here's a thought. When something's called highly selected, it means it's a helpful gene. It's very, very helpful. And in fact, it gets passed to its children because it's probably needed for survival. That's why it's so highly selected. And it brings us to polar bears. They're closely related to the huge brown bear. And as these creatures migrated north, they have clearly incorporated key changes. You got the white fur, you got the ability to eat fat and not suffer for it, which we all envy as humans. And you might ask, well, how do they do that? With some new cool complexity, some new cool genes show up, because we really do want to see new genes pop up and start leading to changes. No, they looked at the top 17 most selected genes, most needed genes, and up to 83% had helpful, positively selected genes, estimated, because they have to estimate how these things impact on computer models, to have at least one damaging mutation. So most of the changes, or most of the top genes that went forward with the polar bear, most had degradative changes to allow it to do that. And so this is not evolution. This is not building something. This is de-evolving. The best genes that it needs are damaged genes, or genes that haven't been changed at all. So this is another example. When we go and look at two things that we know are related, we see these differences. And the same thing with Darwin's finches. This is the very famous finch that he used to help develop his theory on evolution. A key difference in all the finches is their beaks. And it's known now that when there's plenty of food found on the islands, the long beak survives. They can go get the nectar on the flowers. When there's a drought, for example, back in 77, you only get a lot of these, no flowers and a lot of nuts, and these are very hard nuts, and the birds that have the blunt beaks, they thrive. The other ones die, literally die. But not off. I think 80% of them die that year. And so how do you get the blunt beak? Well, it didn't involve a complex ability, to control beak shape. Did that evolve? No, it's two damaging mutations that control beak development. And the long beak guy acquires two mutation, and that blunts the beak. So that, again, whenever the things that get touted as evolution is really de-evolution. And the finch has adapted by de-evolving. The polar bear and the finch is evolution at work. It's just that the full capabilities of evolution haven't been displayed yet. We're not building and building to go from 0 to 100. and even your little puppy, all that artificial breeding that's taken place, all the cute things, the color, the size, the muzzle, even being friendly towards humans, is known to be degradative mutations from the wolf, all damaging things. No one's reported, oh, look how this gene has evolved and is doing something different now. No, it all goes downhill. Just like I said, every time we look, we see something trading inheritance, and the poor dog is not going to go back to be a wolf. Imagine, you'd have to overcome so many obstacles. You could not breed, and I'm going to prove to you in a minute, you cannot breed that chihuahua back to a wolf. Impossible, impossible, and I'll show you. Remember, we saw 2 classes of mutations. I think we can all agree that these ones with massive deletions, they're gone. And these over here, they're degraded with point mutations. Now, surely we can fix these severely damaged ones. Only have a few amino acid changes. Surely we can fix that. If the bug needs them, surely it can fix them. So here's a scenario. Somewhere down the road, Linsky's E. coli escape the lab, and they end up in the local sewer. And now they encounter, say, ribose as a potential food source. Of course, they can't eat that because they jettisoned that machinery, back in the 90s to grow faster. And let's assume that ribose is degraded because of a few amino acid changes. Now, here's your DNA. This is your code. This is your gene. And let's say these are 3 changes right here that knocked out ribose, 99% degraded. And let's say you want to change back and get that ribose. So you fix one of those, an A becomes a T. And now you're waiting for these 2 guys here to get changed. What's more likely to happen? It's random, remember? It's more likely the A is going to switch back to one of the other 3 options. And by the way, to go from A to T, it has to pick between one of 3 options. So there's only a 33% chance it'll pick T and not C or G. And before you know it, while you're waiting for this coordinated change to happen, I say coordinated because timing is everything here, you're going to get 2 other random mutations. These guys pop in. So it's hard. And I'm going to tell you how Lynch approached this problem. And you might argue, okay, Tim, but the genetically devastated E. coli can acquire a new copy of the gene from her new buddies that live with her in the cesspool, the sewer. Okay, maybe. But really all I care about, though, is creating new information. Evolution must build complexity to be creative force that it's claimed to be, vigorously claimed to be. So let's create something new. What's the basic theory? The basic theory is, for example, if a bacteria is copying its DNA, it can accidentally make an extra copy of this citrate promoter. And there it is. You got more DNA. Someone asked before, where does information come from? Well, how do you build? I think the question was, how do you get bigger genomes? Well, it can screw up and bring DNA in. It can get a new virus to pop up and integrate, and you got new DNA. Or it can make its own copy accidentally, and there it is. And so the theory is, this guy's needed, for example. So it maintains that it selects for that guy, but this guy's free to mutate because it doesn't really need it. And let's say these are the 4 changes that are needed, but you need all 4 to change together. And of course, that's hard to do, as I'm going to show you. But if it doesn't get those 4, it doesn't evolve. And I would propose that almost always it needs a lot more than 4 to do something different than what it did before. So let's be generous. But what's most likely to happen is even if this guy gets fixed, it goes from an A to a T, and these guys are waiting for their turn, telling this guy not to change. Hey, buddy, don't change. We got to change. Then we're going to have this activity. No, likely scenario is other random changes, which can happen anywhere in the DNA of this gene. They're going to show up first and hurt the situation. But let's make this really, really simple. Let's just fix 2 spots in the DNA to get our needed change. So Behe asks a simple question. He thought of it, the simplest structure that could still be called irreducible complexity. And he chose disulfide bonds. These are ubiquitous in proteins. They're vital to the structure. To make this thing fold correctly, you got to have them, for the most part. It requires 2 cysteine amino acids plus the general structure of the protein that it's found in. You need those 3 parts. This is our irreducible system we have here. You know, it's like this, it's probably not even true, could be called that, but it's the simplest thing we can think of that has to have coordinated things going on. And what they do, these cysteines, when you have them and they get close enough together, they can bond to each other like this. And it's like a structure. It's like, you know, putting a rod between two of these guys, and it preserves this fold here that you see all over this guy. So they're very, very important in proteins and relatively pretty simple. So how do you test this? Well, you build a model and you estimate how many generations would be required to change 2 specific amino acids to cysteines. Seems pretty simple. You got to make some assumptions in any model. What's the DNA mutation rate? What's the ratio of helpful to harmful mutation should I choose? What's the population size that I will get to play with? Is it 10,000, or is it infinite, which is a common assumption for mathematical reasons? He said, his results said 1 billion generations are required to produce a new feature, and this feature simply has it changing 2 particular amino acid sites, 1 billion generations. Well, what was the response from science? They were outraged. What did we hear? Basically, they heard this. They heard that, you know, if Darwinian processes can't fix 2 measly amino acids in a coordinated fashion, it has no power to create, period. They knew that, you know, let alone 3 or 4, you know, 2. So here's what the math looks like. Roughly, if you're trying to fix 2 spots, it might be 10,000 times 10,000. And that might give you roughly 1 billion. Well, here's the point. If you have 3, then it's 10,000 times 10,000 times 10,000 and on for every additional change. And even this number is just plumb ridiculous. So what does this mean? Well, in human terms, you know, 1 billion generations is 20 billion years, given a conservative 20-year per generation number. The universe is only 15 billion years old, we're told. So this whole concept here, you know, makes the upward process of evolution seem silly to even begin to talk about it, because this is devastating if it's true. And go ahead.
So let's wait for questions until the whole thing is done, okay? So we can just get through the lesson.
So Behe's paper never claimed evolution could happen. Just that one particular very popular model was implausible. But science says, and not so fast, Behe, we see what you're doing here. So there was a lot of commotion. The paper was ridiculed for allowing publication by a known intelligent design advocate. You know, how dare you? Within a week, he was told, you know, none other than Michael Lynch, the very famous mathematical geneticist who popularized neutral mutations as a force, he's going to write a response to your paper, which is almost unheard of, you know, in publications, but it happens. And so, of course, Lynch took his model, his own model. He made some generous assumptions, more than Behe. Okay, not a problem. What was his result? No, he said, Behe, you're wrong. It's not 1 billion generations that's needed. It's only 100 million generations. Only 100 million generations. And I said, oh, is that all? You know, in human years, that's only 2 billion years of intense evolution, and you get 2 specific amino acid changes. What? Really? 2? You know, I don't understand why Lynch even said this out loud, you know. Just because you drop one of the zeros here and you get 100 million, that's more plausible. Let me explain what that means. We are told, you know, that we evolved from humans, right? We're told that, you know, how, you know, you look at a chimp, you look at a human, you can see how easily that could happen if you think about it. They say our DNA is like 96, maybe 99% identical. You know, surely by just looking at the 2 creatures, one can see how easily we could transform from a chimp. Now, the common number given for that evolution from chimp to human is 4 million years, which is a pretty short wink of an eye from the evolutionary standpoint. And I say 98%, wow, you know, what does that really mean? And of course, I use my maxim is that evolution makes a lot of sense until you stop and think about it. And now we're going to stop and think about it. Humans have 3 billion pairs of DNA, 3 billion. So a 2% difference means there are 60 million different DNA base pair changes, 60 million. Now, so let's say it's only 1%. Okay, so that's 30 million changes, you know, whatever. I call that great marketing, because I watch these commercials and they'll say, our lunch meat is 97% fat-free, which sounds really impressive. What they're not saying is, hey, our meat contains 3% fat. Hey, we're 99% identical to a chimp. Wow, that's crazy. We're so close. Flip side of that coin, there are 30 million known DNA base pair differences between us and the chimp. That doesn't sound quite as catchy, does it? And in case you're missing the point, 30 million is bigger than 2. So evolution says, give me 4 million years, and I'll give you 30 million changes, and I'll make you a human from a chimp. Okay. Our friend Lynch says, give me 2 billion years, which is a lot longer, and I'll give you 2 changes. Or maybe it's 2 to 6. I don't know how he did his actual model, but who cares? It's 6. Okay, fine. And this brings in the comparison. This is called the principle of comparative anatomy, that I think is missing in a lot of the thinking here. Thinking goes like this: if this is kind of hard to do, 2 billion years, if that's kind of hard, this is crazy hard. Okay, so I tell people, stop trying to imagine what's in the middle and washing it away. We can't even do something simple, and you're discounting something extremely hard. You got to think about this. So there's no way these bacteria cannot go home again. If you need 4 changes happen, this is the likely scenario. And you can't even fix 2 spots. And so Darwin might object. He says, Tim, forget humans, forget the 20-year generation. Let's focus on bacteria. They can double every 20 minutes, and they can. That's why I like working with them. They're fast. Now, that means 100 million generations per Dr. Lynch for bacteria just to get 2 specific amino acid changes in a protein. We just looked at Linsky. He did 50,000 generations, which is a lot less than 100 million. And what did we get? We got chaos and destruction. As Darwin swooped in and helped, and I use that term loosely, he helped our friends trapped in the flash to grow faster. So a new change in the environment occurred. Okay, that was this Linsky experiment, and that was the Darwin pressure. It came in, and to help our friends, Darwin rushed in with degradative but beneficial mutations. Thank you very much, Mr. Darwin. And I say, Dr. Darwin, why did you not bring some nice new complexity? Like a gene that speeds up replication. A new gene that does that would be really cool. And he would say to me, well, Tim, that's a bit harder to come by. Just to change 2 amino acids needs 100 million generations. And besides, I fixed your growth problem in way under 50,000 generations. Stop your complaining. I did my job. So now we're getting a new vantage point. And this is hot. I mean, all this information is very recent. We're still digesting it. And yes, evolution is real. It's powerful. It's just not as powerful as had been ascribed. It has issues generating complexity. It rushes in to help to adjust to our changed environment. Something changes, it comes in and says, how can I help? It will randomly test every possible amino acid switch in your proteins long before it can do any kind of coordinated 10 or 5 or 2 specific changes that would be needed to generate an instant change. So the general thought we have is this. People think, well, we got all the time in the world for Darwin to do its thing. 15 billion years, my goodness, that's a long time. It's like whenever it's needed, we can summon up some spirit of molecular complexity and it'll come to fix what ails us. Well, the new reality is to alleviate new selective pressure, every degradative change, every damaging single-step mutation would need to be tested multiple times, or would be tested multiple times, not need be, it would be tested, as a solution to your problem, eons before a complex solution could be tested. And so everyone agrees evolution is blind and it's an aimless process. But now that genetic evidence is rolling in fast, we are seeing exactly what one expects from a blind, aimless process. Last week we asked, where's the fossils? Why is it missing? Well, now we know it never existed. Lynch told us 100 million generations gives us 2 lousy amino acid changes. If true, we could never have evolved from chimps. Nothing could have evolved, actually, upwards. And this is the second law of thermo. It's vindicated. Things go downhill. We're wound up, we go downhill. There's nothing to stop it. So realize this, if you're an evolutionist who denies God's existence, denies, the latest molecular genetic evidence says you have a lot of faith, okay? And that's why I entitled this, Evolution, an Encounter with Faith. So, folks, keep the faith, and but be reasonable about it, okay? Thanks a lot, guys.